Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your aseptic techniques. Okay, we all know that this what is this technique means. Simply the aseptic technique means that the process which we do in a sterile condition. Okay, process that we perform under that sterile condition. Now what is this sterile condition? So the sterile condition are simply means that free from any type of microorganisms. Okay, we all know that this microorganisms are what, either they are spoilage or either they are what, they are pathogenic. Okay, now these pathogenic are very they are very concerned because it cause disease. Okay, so when we perform the when we give a sterile condition, then these microorganisms cannot be grow. So simply the aseptic techniques are what they are, process which we perform under a sterile condition. These aseptic techniques are used in different laboratories like your cultural laboratories, human laboratories, even tissue transplantation, and these sterile condition are also performed in a microbial laboratory. Now see what is written in the slide. Aseptic technique refer to a procedure that is performed under a sterile condition, the condition which are free from any type of the microorganism, which includes medical and laboratory techniques which deal with culture and human cells and tissue for transplantation. See, the medical laboratories are required the sterile condition. Why they are required? Because if any tissue are transplant, so at that case the environment should be sterile, otherwise the microorganism will grow and it will damage the tissue. That's why the sterile condition are very important. Now come to the next slide. What is the aim of the sterile technique? So the main aim of the sterile technique is what? We have to prevent the growth of the microorganisms. Okay, suppose that when we are working under a micro lab. Now, in micro lab, suppose that we have to grow a bacteria A. Now, this A bacteria can be grow under a sterile condition, which means that at that condition in which the other type of the microorganism will not grow. If we are not providing this sterile condition, then uh, along with this A bacteria, the other bacteria like your B, C, D will also grow, and this B, C, D will contaminate the test or preparation or media. So what we have to do, we have to prevent the growth of the microorganism. To prevent the access of microorganisms during the preparation and testing. Okay, so what we have to do, we have to prevent the growth of the microorganisms during the preparation of the media, and even also when we perform a test, at that time also we have to prevent the access of the microorganisms. So this is the main aim of the aseptic technique. Now the next is in the microbiology lab we use aseptic technique that I already told you. So we have to prevent the contamination of the specific microorganism we are working. Suppose that we have to grow a bacillus. Okay, now if we do not provide this sterile condition along with bacillus, the other microorganism will also grow. So when we give a sterile condition, then at that case only the bacillus microorganism will be grow, and it also prevent the contamination of the room and personal with the microorganism we are working with. Okay, so when we provide a sterile condition or aseptic condition, it also prevent the contamination occur by the room and by the environment and by the person, suppose that who are working at that particular lab, so that this microorganisms can easily work. So these are the main aim of the microbiological lab when we are using the aseptic technique. Now see what is written here: prevent contamination of the specific microorganisms we are working with, prevent contamination of the room and personnel with the microorganisms we are working with. Now the next is your sources of the contamination. Now what is this contamination means? See, the contamination are simply means that the presence of the impurities, or the the ingredients which are not the part of the that particular system will added into that system either intentionally or either intentionally. Suppose that we have a nutrient agar media. Now we all know that this nutrient agar media contain a beef extract, sodium chloride, okay, along with that agar and that peptone. Okay, now like these are the composition of the nutrient agar media. Now apart from these component, some other component will be added into a nutrient agar media, will consider as a contamination. Now here the contamination are what? Contamination are considered as a introduction of the microorganism into the media, or testing. These are what? These are called a contamination. Now there are different sources of the contamination, because we all know that the microorganisms are present everywhere. The microorganisms are present everywhere, like your atmosphere, breathe, hand, cloth, hair, working surface, equipment. So they are what? They are become the sources of the contamination, because they here the microorganisms are present everywhere, and from that sources they will enter into the interfere into the testing and also interfere into the media. So these are what? These are the sources of the contamination. Now come to the next slide. Some terms that we have to discuss when we are discussing the aseptic techniques. Now these terms are differentiate from each other, but when we hear these words we thought that these words are very similar. Now the first word is your sterilization. So this sterilization are simply a process in which the particular media or the particular surface are free from all type of the microorganisms. Okay, in that case there is there is no chance of the growth of the microorganisms, and even the microorganism which are present at that particular area will also kill. Okay, like all types of the microorganisms, like your spore type, vegetative cells, all type of the microorganisms are killed when we perform a sterilization. Now sterilization is a process by which article, surface or medium, see here we consider all these things like articles, surfaces or medium, is made free from all microorganisms either in the vegetated or spore state. Now disinfection is another term in which the pathogenic microorganisms will kill. Okay, so pathogenic microorganisms are what? Which have a capability to cause a disease into a human being. So in disinfection what we have to do, that particular surface, the particular area will be free from all type of the pathogenic microorganisms. Now see what is written here. Disinfection is the process by which an article, surface or medium is made free from all pathogenic microorganism, the microorganism which have a capability of giving rise to a infection, means that the microorganism which have a capability to cause a disease into a human being. Okay, now the next is your antisepsis. So antisepsis is another term that we frequently use when we are discussing that aseptic technique, which means that the particular process in which the growth of the microorganisms are inhibited. Suppose that in a particular area, like three five microorganisms are present. Now this five microorganism will not kill when we are performing the antisepsis, but their growth will be restricted, which means that when we perform a antisepsis, the microorganisms undergo a sleeping condition. They will sleep, but does not kill. When antiseptic condition are removed, again they will grow, and they will capable for causing the different type of the infections. Now what is written here, it is a process by which the growth of the bacteria are inhibited, but they are not killed. That I already told you. Now what need to be sterile in the process of aseptic technique when we are performing a aseptic techniques? Okay, so like when we consider aseptic techniques, not now what type of the instrument, what type of the glassware we have to sterilize. Again here sterilization are what? Sterilization means that the particular surface or area should be free from all type of microorganism. Okay, so in that case we have to sterilize the culture media. Culture media means that the media which is used for the growth of that particular microorganisms. Okay, and the second thing that we have to sterilize is a fluid which is used in a lab, like your distilled water. In case of the distilled water, what we have to do, we have to use a sterile distilled water. Reagent should also be sterile. Laboratory containers should also be sterile, and laboratory equipment. Laboratory container means that the Petri plates, the test tubes, beakers, all things should be sterile. Laboratory equipments like your laminar airflow, because we have to perform the experiment inside the laminar airflow, and how we sterile all these things. The video related to this are already uploaded in our account. You can visit our account and see that videos. Okay, now these are what? These are the things that we have to sterilize when we perform a, or you can say that during the aseptic, during the process of aseptic technique. Now method of the sterilization. So here the first method of the sterilization is what? Physical heat. This physical heat are divided into a two part. One is your dry heat and second is your moist heat. Dry heat will be performed two under two instruments. One is your hot air oven and another one is your flaming. Okay, so in hot air oven what we have to do, we sterilize the glassware. Glassware should be sterile at 150 degrees Celsius for 30 minutes. For 30 minutes. Okay, and flaming, we sterile the inoculation loop and needle. When we have to perform a streaking, we need an inoculation loop, and this inoculation loop should be sterile by using a flame. Okay, now when we sterile the glassware with hot air oven or inoculation loop with flame, we use a dry heat. Now moist heat means that when we sterile under a high pressure, then we required what? We required a steam. So this will be done inside the autoclave, and in autoclave we sterilize the culture media. Now the next is your filtration. So this filtration are used in laminar airflow. Laminar airflow, in which what we have to do, we like we use a HEPA filter. We use a HEPA filter which does not allow even the smallest microorganisms to pass through that filter. Okay, so this is the use of the filter, which is used for a heat sensitive liquids like antibiotic solutions and sera. And we also do a filtration when we have a liquid which are what, which are very heat sensitive. So in that case we also use a filtration like antibiotic solution and sera. Radiation will be used for enclosed areas like inoculation room and viruses lab. Okay, this can also be used like your laminar airflow when we switch on the UV visible light. Now the next is your chemical methods, like with the help of the alcohols and the aldehydes. These alcohols are used for the sterilization of the surface, like when we sterilize the surfaces of the laminar airflow, and also it is used for the sterilization of the skin. Like we can use a 70% of the ethyl alcohol uh, for the sterilization of the hand. In aldehyde, we use a formaldehyde, which is used for the sterilization of the equipment like your centrifuge. When we have to sterilize this centrifuge, then we use what? We use a formaldehyde, which is what? Which is a aldehyde. Now the next is your phenol. Phenolics like your phenols, which are used for the sterilization of the floor, wall, and benches. Okay, in that case we use what? We use a phenols. Now the next is your general principles of the aseptic technique. Disinfect the work area before starting to reduce a potential contaminant on the bench top, and after work is finished to protect other from a possible contamination. Okay, so the first principle of the aseptic technique is what? We have to sterilize the work area or a bench before starting the process and when we finish the process. Now why we have to disinfect that work area before starting the process? So that we reduce the contaminant on that bench top. We all know that when we perform a that when we perform when we perform an experiment under our laminar air flow, in that case what we have to do, we clean the laminar flow with the help of the 70% of the ethyl alcohol before starting the work, so that it will reduces the chances of the contamination. And we have also cleaned that benches after the experiment, so that if any contamination are there, it will not pass through the other person. So to protect the other person from the possible contamination, we have to clean off when we finish the work. Now the next is your flame. Flame the inoculating loop before and after making a transfer of the bacteria from one container to another. We all know that when we have to transfer that culture from one container to the another container, we use a inoculation loop, and we have to sterilize that inoculation loop by using a flame before transferring and after transferring. Before transferring, so that we avoid the contamination, we avoid the transformation of the contamination, and after transfer, so that it will not contaminate that particular other things. Never lay a inoculating loop on a bench top if you are not sure it has been flamed first. When in doubt, flame the loop again. Okay, so this is what? This is a sterilization of the inoculating loop when we are performing the experiment. Now the next is your flame the opening of the glass container before removing the bacteria from them, and again after bacteria have been removed. Likewise, flame the opening before transferring bacteria to a container, and again after transfer is completed. So what we have to do? We have to flame our container, glass container in which the culture are there when we are transforming in, or even when after transferring, we have to again flame it with the help of that flame. Do not lay the cap of the container of the bacteria on the bench top while bacteria are removed from or transferred to the container. The cap should be remain under your control throughout the transfer. Okay, so we all know that when we are performing the experiment, what we have to do, we put the, like sometimes we do a mistake, we put the cap of that container, either the Petri plate or um, either the test tube, like we put that cap into the bench. So this is not a right thing. Okay, we have to control that cap throughout the transfer. This is not the right thing, that we have put that contain cap into the bench. Work quickly and efficiently to minimize the time the culture is exposed to the environment. When we are performing the, like when we are transferring the microorganism from one container to the another container, we have to do this work very quickly, so that it will minimize that the exposure time to the environment. Example of a transfer, typical transfer. As an as an example, there are the step which is required for a good sterile technique in transferring bacteria for one cap test to another. Flame the inoculating loop, flame the mouth of the tube while holding the cap, remove the some bacteria with the loop, flame the mouth of the tube and replace the cap, flame the mouth of the tube which the bacteria are being transferred, inoculate the tube with the bacteria from the loop, flame the mouth of the tube and replace the cap, flame the inoculating loop. Now this is a way for transferring the bacteria from one container into the another container. This is a like representation of with the help of that images. Okay, so this is all about the aseptic technique. I hope you will understand, but if you have any query related to this topic, you are free to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also visit our website, that is www.itlsacademy.com, and ask your query to the helpline number that are already mentioned in your slide, 7080833450. Thank you for watching. Please like, share and subscribe our channel.